We're going to go ahead and pick up with the next chapter, chapter 6 here. So we're going to be talking about the cell cycle. So it's the life cycle of a generalized cell. So it's not really a specific type of cell that we're going to be looking at. It's more kind of the general steps that each cell would go through. So the cell cycle is split up into several distinct phases. So these phases are split into different periods of growth. They're split into reproduction and also the normal functions of the cell. So it has four main stages. Okay, so it has G1 phase, which is gap 1, also oftentimes is referred to as the first growth period. Then you have the synthesis phase. So in this step, they are synthesizing new DNA, so they're copying all of their DNA, getting ready for division, cell division. Then you also have a secondary growth period. So this one's called gap 2. And this is where it's kind of going through its final check systems to make sure that everything is ready in order to split the cell in half and to duplicate itself. And then that process of actually splitting itself in half is called mitosis, or the M phase. So here you've got interphase. It's really can be split from those four phases. It can actually be grouped into two main stages. You've got interphase, which includes both gap 1, synthesis, and gap 2. And then you also have mitosis, which is the actual cell division stage. Okay. So again, gap 1, you're going to have cells growing, carrying out their normal functions, replicating their organelles, just kind of existing and completing their purpose. Then you have your synthesis stage where you're going to copy the DNA, and then go straight into gap 2, which is your additional growth period. Starts right after DNA is finished being replicated, and then contains those final checks to make sure that the cell can properly divide and that it's ready to divide. And then later on, we're going to go into the different phases of actual mitosis, the cell division stage. So in gap 1, again, you're going to see cell growth. Basically, gap 1 starts right after a cell divides. So after it divides, it's at its smallest size. So it has to go through a lot of growth in order to be ready to divide again. So that's why this step, if you look in this little chart here, this step is the biggest step of the cell cycle. You're going to have normal functions, so we've been talking a lot about those. You're going to have a lot of ATP synthesis using cellular respiration, and really just kind of going through the motions of what that cell is designed to do. And then it's also really just recovering from that previous division. So it's creating more cytoplasm, it's replicating its organelles so that it's actually up to full capacity and can really do everything it needs to. And it starts to prepare for the next division. So it's kind of, the cell divides and then immediately starts getting ready for the next division. And then it goes into synthesis. So in synthesis, DNA is copied. And saying that phrase, it sounds really simple, but it's actually a relatively complex process, which we're going to get into later. DNA does not actually condense during this stage. So what you'll see in the cells is that DNA is just kind of this tangled mass of nucleotides that are combined together, and it's separated into what are known as chromosomes. So humans have 46 chromosomes, but when it says condense, that whole tangled mass of DNA is really great when you are trying to go in and read a certain gene or use the DNA for its purpose. But when you're trying to separate the cell and make sure that you have a copy of each chromosome in each of the new cells, that tangled mass of basically string is what it looks like, is awful. Okay, so before the cell actually divides, it has to condense its DNA. And so if you've ever seen a picture of a chromosome, generally you see it kind of looks like a wonky looking X, that's the condensed DNA. So it condenses down into these very solid, visible chromosomes that do look like X's. And that makes it really easy to deal with, to divide it and make sure that each cell has its own copy of every chromosome. So then it enters the gap2 phase. So you're going to get additional growth, and it goes through its final preparations before division. So it's kind of like the system checks that a pilot would go through before they take off. So they have to make sure, you've got enough gas to reach the destination, have to make sure that there are no incoming planes, that you're not going to hit a plane on the runway, all these different kind of checks that it has to go through, or that the pilot has to go through before they can actually take to the air. So the cell has its own version of those kind of final checks before it goes through division. Because division is kind of, it's a very energy dependent process. And so if the cell is not actually ready to go through division, it's better to know that before it starts than to get halfway through, run out of energy, and then have to kind of start all over. And then it's got like the halfway divided cell, and then it's going to divide that again, and things get all screwed up. So it's better to know beforehand that it can actually go through the whole division process and really complete it perfectly, or hopefully perfectly, before it gets started. So it also checks to make sure that the DNA is duplicated and that it is undamaged. Okay, so that's a big one. The process of DNA replication, that again we're going to get into later, has a bunch of checks and balances in it to make sure that the DNA is being duplicated properly and that it's not damaged. This phase is also when the cell can basically decide that, okay, the DNA is way too damaged for me to really divide and create another cell that has this damaged DNA. So the cells actually have something called apoptosis, which is programmed cell death. And so if the cell determines that its DNA is way too damaged to actually function properly anymore, it will undergo apoptosis and basically just take itself out so that its damaged DNA doesn't get passed on. So it also double checks the size of the cell. So because it's taking a large cell and splitting it in half, it actually has to be large enough that when it's split in half and separated, the two cells that are created can actually continue with their cellular processes. Okay, because if those cells are too small to do anything, then there's really no point in dividing and making those really tiny cells, because then they'll just die off anyway. Alright, and then you get into mitosis. So mitosis is just a fancy word for cell division. Okay, so it is divided into several distinct phases. So you have prophase, metaphase, and then it goes into anaphase, telophase, and cytokinesis. So cytokinesis marks the end of mitosis. So it is actually when the cells kind of pinch off and go from one cell into two separate cells. So we're going to go more into depth in each of these phases later on in this chapter. So we're just going to leave it at the names right now. But easy way to remember the order that I've always found is PMAT. So P, prophase, M, metaphase, A, anaphase, T, telophase. PMAT. Okay, so depending on what type of cell you have, they are actually, or what type of cell you're looking at, they are actually going to divide at different rates. Okay, so the type of cells that get damaged much more quickly, have, different kinds of radiation that they hit. They're designed to protect the rest of the body from something, or they're in contact with a really, nasty environment of some kind. They're going to have to replicate much faster than the type of cells that are kind of just hanging out inside the body, doing their function, but they don't really get damaged much. So if you look at this, you can tell just based on the number of days that the intestines, the internal lining, has the roughest environment. Okay, so if you think about that, that includes the lining of the stomach. So we know that the stomach produces stomach acid in order to help break down the food. Well, that is a really tough environment for any living cell to exist in. Because if it gets, if the outside of the cell membrane gets broken down enough, that acid can actually get inside the cell and start wreaking havoc. So these cells actually produce a mucus coating that coats the outside of the cells to help protect them more from the acid, but they do still have to be replicated every 4 to 5 days. Okay, so there's a lot of growth involved with these cells, and then there is a lot of energy needed simply because they're replicating at such a quick rate. Then you've also got skin cells. So skin cells will replicate roughly every 2 weeks. Okay, and if you think about that, that is because they are in contact with the environment outside of your body. So they get kind of beat up quite a bit as well. They're exposed to UV radiation from the sun, and then they're exposed to anything and everything that you touch throughout the day. So that can cause more damage over time. So they still have a relatively short replication cycle. Then you've got red blood cells, liver cells, and then at the top there, at the bottom, but you've got the intestines, the muscle, and other tissues. So on the other side of the internal lining, that takes about 16 years before they replicate. So those obviously don't get damaged much, and they have a relatively slow rate of growth, simply because you wouldn't want them to grow constantly for 16 years. It would end up being massive. And, cells, they have kind of an upper limit of how big they can be. So on average, the cells in the human body are between 7 and 10 years old. So there is kind of one of those weird little sayings that you're a completely new person every 10 to 20 years. Okay, it means that every cell in your body has been replicated. It's a new cell every 10 to 20 years. Okay, so it's kind of a weird thing, but some cells are much younger, obviously, because some of them get replicated every 4 to 5 days. Many other cells are much older. So cells like bone cells, muscle, or neuron cells, so your brain cells, don't replicate very much, if at all. Okay, so brain cells are specifically ones that don't replicate very often, if at all. And that's why any kind of brain damage is such a major issue. It's because your body doesn't have a way to heal that. They don't replicate those cells. So once those cells are gone, you're kind of, SOL. Sorry, out of luck. So some cells are unlikely ever to divide. They are in what is known as G0 phase, so it's a constant growth and normal process type phase. Those are neuron cells, so what I was just talking about there. And so a lot of research has been going into trying to grow neuron cells outside of the body so that they can be implanted into patients who have any kind of brain damage. So that includes like your brain stem that goes down along your spine. So that's why spinal injuries or head injuries are such a big deal, because if those neurons are severed or damaged, that can lead to paralyzation. Okay, so you become paralyzed from that spot down. Muscle cells also don't really like to divide. They will grow quite a bit, especially if you are working on growing them. All right, so we are going to stop there on this video and pick up with cell size in the next video.